Hello and Assalamualaikum everyone. My name is Iza and meet our team members.
Hello, my name is Nurina Bila binti Ustazah.
Hi, my name is Shima.
Hi, I am Nini.
Hello, my name is Hazima.
In this video, we would like to show you two experiments that are, the first one is home DNA extraction and the second one is investigating light as a necessity for photosynthesis. And these are the contents that will be discussed for each experiment. And without further ado, let's just get into it.
For the first experiment, we wanted to extract the DNA from a banana simply by using the ingredients that we can get from our kitchen. At the same time, we wanted to determine whether different amounts of alcohol added could affect the net amount of DNA obtained. Before we begin, let's get to know how DNA looks like and what is DNA's purpose to living beings. DNA stands for deoxyribonucleic acid, which is a molecule that contains an instruction that every living organism needs to develop, live, and reproduce. DNA is located in the cell nucleus where it is called nuclear DNA. DNA is a molecule composed of two polynucleotide chains that coil around each other to form a double helix that carries genetic instructions in living organisms.
DNA extraction is the process to purify DNA by using chemical method to separate DNA from the cell membranes, proteins, and other cellular components.
This process is carried out by breaking down the cells to release DNA, separating DNA from the proteins, and precipitating them.
Now we are going to show you how this experiment is conducted. Without further ado, let's just get into it. These are the ingredients that will be used for this experiment. First, weight the banana and make it as the constant variable for two experiment setups. Next, the previous banana is added into the Ziploc plastic bag. Then, remove all the air that was trapped inside the Ziploc bag before sealing it tightly. To physically break down the cell wall, crush the bananas until they become into smaller pieces and slightly become liquidy like this. By using measuring cups, add 1 per 3 cup of water, 1 tablespoon of detergent and 1 per 2 teaspoon of salt, and then mix all of the ingredients together in one container. This solution will be used as the extraction solution or buffer. And by using measuring cups, add 3 tablespoons of extraction solution into the previously crushed bananas. As always, seal the bag properly before mixing all of the ingredients. And please ensure that all of the ingredients are mixing together. And it should look like this. Next, take the previous mixture and filter them twice. This is to make sure that all the debris or any impurities are filtered out from the mixture. By using a spoon, slightly press the leftover above the filter to add some force while filtering out the mixture, and repeat the same step for the second time filtering out the mixture. After that, alcohol with 75% concentration is used for 2 sets of experiment with different amount of alcohol. And then, add the alcohol into the DNA extraction solution by slowly pouring along the wall of the container. This is to prevent the DNA extraction solution from mixing up together and disrupting the DNA from the living organism. After that, leave the solution for 5 minutes and we will see the result. With the usage of 1 per 4 cup of alcohol, we managed to obtain this result. And as you can see here, we managed to obtain DNA from bananas that appear to be a white stringy layer, even after leaving it for 5 minutes. We also leave the solution for 1 day to ensure the white stringy layer is more clumped and easy to handle before weighing process. Meanwhile, with the usage of 1 per 2 cup of alcohol, we managed to obtain this result, which the stringy layer looks more abundant and more DNA is obtained. But why this happened? This is because the DNA is not soluble in the alcohol, but it is soluble in the water. The alcohol, or to be specific, isopropanol, is miscible with water. Hence, the more alcohol is added, the solubility of DNA decreases. And finally, to prove our observation, we carefully weigh down the stringy white layer to determine the net amount of DNA. And let's see the result.
The result shows that the net amount of DNA obtained with the usage of 1/2 cup of alcohol is higher. This shows that by increasing the amount of alcohol, the net amount of DNA obtained also increases. The basic principle of DNA extraction is disruption of the cell wall, cell membrane, and nuclear membrane to release the highly intact DNA, followed by precipitation of DNA and removal of the contaminating biomolecules.
Just like us, bananas also have DNA in their cells to determine their traits. In this activity, we use banana as our chosen living organisms.
In this experiment, we use common household products such as salt, detergent, water as DNA extraction buffer, and alcohol to break apart the cells in the bananas and extract out the DNA. The function of each of the ingredients are listed as shown here.
To conclude, we are able to obtain DNA extraction and identify the optimized method by manipulating the variables involved.
Now, let's move on to the second experiment where we wanted to investigate how the presence of light could influence the production of oxygen as one of the photosynthesis products. Enough for the long wait. Let's just jump straight into it.
Hi, now we proceed to experiment 2. As you can see, these are the materials that we need in this experiment. First, place a leaf in a glass. Fill the glass with water and let the leaf submerge in water. Label the glass as A. Put glass A under the sunlight and leave it for 3 hours. Perform the same step to the second leaf and label it as B. After that, put glass B in a room where there is no sunlight and also leave it for 3 hours. Finally, after 3 hours, we can see the difference between both leaves. There are bubbles around leaf in a glass A, while leaf in a glass B is clear without bubbles.
Based on our results, the leaf in glass A, which has been exposed to sunlight, has bubbles surrounding the leaf. This shows that the photosynthesis process did take place in leaf A. But how? Let's do a quick review. To explain this situation, bubbles that we see just now are oxygen gases. In general, we know that during the photosynthesis process, oxygen gases are produced along with energy in the form of carbohydrate and of course with the help of solar energy, carbon dioxide, and water supply.
The real question is, how does the oxygen is produced? Oxygen is produced as a waste product during the photolysis process, which occurs in a light-dependent reaction. Bear in mind, there are 2 photosynthesis stages. One is the light-dependent reaction, which requires light to occur, and the other one is a light-independent reaction, or known as Calvin cycle, which can work either in the presence or the absence of light. Back to our focus on the light-dependent reaction. Where does the photolysis process take place? When light energy is captured by the 2 photosystems, photosystem 2 and photosystem 1, the light energy is transferred to a chlorophyll reaction center, causing the electrons in the reaction center to become energized and escape through the electron transport chain.
To replace the electrons that have escaped, this is when photolysis occurs, where the water is split, releasing 2 hydrogen ions, 2 electrons, and oxygen gases. Keep in mind that all of this process takes place in the thylakoid membrane of the chloroplast in the plant cell. These processes only happen with the presence of light.
Without any sort of light, the light-dependent reaction cannot take place as well as the photolysis process. Therefore, no oxygen gases are produced and no formation of bubbles is observed, just like we see in the leaf in class B, which was placed in a dark room with no source of light.
To sum up, we can say that photosynthesis takes place with the presence of light, especially during light-dependent reaction. At the end of this experiment, we are able to deepen our knowledge regarding the processes involved during photosynthesis as well as identify their products.
And that is all from us. Thank you for watching.
Bye!